Hi folks, in this video we're going to go over two different types of cell division. The first is going to be how do you just replace the cells in your body that are dying, whether it's the cells of your skin, cells of your stomach lining, and then in contrast we're going to look at how you make gametes known as sperm and egg. And this is going to be a completely different type of cell division. So if you're just replicating cells, you're going to do something called mitosis. You have learned about this before. And the new type of cell division we're going to talk about afterwards is called meiosis. So regardless of which you're doing, we're going to start with a typical cell in your body. Now, whether this cell is in a testis or an ovary or your skin or your stomach, it's still going to have two full sets of chromosomes, and we're going to call that diploid. So if you don't remember that term, diploid means two sets of chromosomes. For every size or type of chromosome, you got one from a mom, one from your dad. Okay, another way of depicting this is to put the word or the term 2N. That indicates two sets of chromosomes, not 1N, not 4N, two sets of chromosomes. And because neither you nor I want to have 46 chromosomes shown here, the cell I'm using here only has 4 chromosomes total, so two sets of 2 chromosomes total. Now, I just remember that when we're talking about humans, it should be 2n equals 46, not 4. Okay, regardless of if you're going to do mitosis or meiosis, whatever you're going to do, the first thing you're going to have to do after growing the cell a little bit bigger is to replicate those chromosomes. So that's S phase, and by the time you're done with that, every chromosome is going to have an identical replica with it. Those two are called sister chromatids. We'll come back to that a little bit later. But this is the sort of point where you can go one or the other way. If you do a mitotic cell division, and that's what we're talking about now, mitosis, you enter M phase, and you line up all of these replicated chromosomes individually, and you just yank them apart. All right, we use motors to pull these two identical sister chromatids apart. And in the end, the cool thing about this is you're just going to have two identical cells. You replicate the cell that you started with, and now you have an identical copy. That's the purpose of mitosis. You replicate a cell, replace a dead cell, or just allow the organism to grow. Okay, now let's compare that to the other type of cell division that we're going into. A meiotic cell division cannot be the same. And that's because what we're trying to build here is to build sperm and eggs. Now, we cannot have two identical cells here. The sperm cannot be the same as the original cell. The egg cannot be the same as the cell that it came from because of the number of chromosomes. So if a sperm had like 4, like we started with in this example, and an egg had 4, then the next generation, the kids, would have twice the number of chromosomes per cell as the original organism. That doesn't work. If we were talking about humans, if you made, again, a sperm and an egg by mitosis, they'd each have 46 chromosomes, and the next generation of humans would have 92. That does not work. That is not functional in most organisms on Earth. So, instead, what we need to do is cut the number of chromosomes in half. We need to pass on one of every, what's called homologous pair, one of each size or type of chromosome, not both. So for humans, that's going to be 23 chromosomes in each of the sperm and the egg, resulting in, back to normal, a kid, perhaps, that has 46 chromosomes. So that's the end goal. So the way we're going to do that, we're still starting with that cell, in this case, that has 4 chromosomes, 2 sets. We replicate those chromosomes, but now we're going to line them up a little bit differently. Instead of lining them up so you can separate the two identical sister chromatids, the two replicas, we're actually going to separate the two what are called homologs, the two similar but not identical versions of a chromosome. And if you haven't followed this yet, the green and the blue are not identical. The green and the blue are similar. They're the same chromosome size, for example, but they don't have necessarily all the same alleles. So they might not have the same exact versions of each chromosome. So what we're going to do is we're going to yank the green and the blue apart from each other. Maybe the green is the one you got from your mom and the blue is the one you got from your dad. You're not going to pass both on to your sperm and your eggs. You're going to pass one. By the time we get to the sperm and the eggs, here's the next major difference. You actually have to divide a second time. Remember, we're starting with, in a human, 46. We replicate up to 92 chromatids. And now, if you want to get down to 23, you've got to divide twice. All right? So 2 separate division steps to make these sperm or eggs. Okay. Now, what you'll notice is these gametes, the sperm in this case, are not the same as the cell that they came from. That is hugely different from a mitotic or mitosis situation. Okay, so we have our 2 divisions, meiosis 1 and meiosis 2 are how they're named, and that gets us to half the chromosomes we started with. Now here's the other thing that you may not have noticed. Each of the sperm is going to end up being unique from each other sperm. Each egg is going to be unique compared to each other egg, and there are 2 reasons for that. Now, the first one I haven't even shown you yet. Neither have I shown you the second version. So here's the first one. One of the things that's worth noting is, notice how I arranged the greens on the left and the blues on the right? They didn't have to be that way. It is entirely random which orientation they are, meaning whether blue goes left or right, green goes left or right. It's randomized. So it's entirely possible that the cell on the left will get little green and big blue as opposed to little green, big green, I guess, based on the way it's drawn there. So this is the next level. This sort of randomization of which way the homologous chromosomes go is called independent assortment. This is, again, separating those similar but not identical versions of a chromosome in different orientations. So you could pass on your mom's chromosome 1 and your dad's chromosome 2, or you could pass on the chromosome 2 you got from mom and the chromosome 1 you got from dad, or any of those combinations beyond that. Okay. So in the end, you'd end up with this next set of sperm. That's just as likely. There's one other step, and this is probably the craziest of all the steps in meiosis that we're going to discuss. And this is the other reason that every sperm and every egg that is produced in your body is 100% unique. There's no way you can make the same sperm or egg twice. And I've just drawn it in. And I'm going to show it again. And if you pay attention, you'll see it happen. And here's a third example of it. What happens during early meiosis 1, before you even do the independent assortment, before you pull apart those homologs, you are actually going to switch portions of the chromosomes. You're going to swap between the 2 versions of each chromosome that you have. So in the end, the chromosomes that you pass on to your kids are not even the same chromosomes you got from your mom and your dad. They are really not. The chromosome 1 you pass on to your kids is a mismatch of the chromosome 1s you got from your 2 parents. This process is called crossing over, also known as recombination, and this is the first of those 2 steps that ensures that every gamete you make, whether it's a sperm or an egg, is completely unique and can't be built again. So I've just shown you what the next part of that meiosis would look like in terms of the chromosome separation, and let me go to the sperm themselves. You can see now, if you include crossing over, each and every sperm really is unique in this case. I'm not drawing this for the eggs because that would be a little bit awkward in terms of sizing on the screen. It's also worth knowing eggs tend to be much larger, and instead of producing 4, you typically produce 1. The other cells are tiny and end up being absorbed. But that's not something we want to focus on a lot. In general, what I want you to see is that instead of replicating 1 cell that's diploid into 2 identical diploid cells, here we're producing unique cells. And these sperm and eggs, these gametes, only have 1 set of chromosomes. That's the big picture for today. Keep in mind, again, we're cutting the number of chromosomes half. We're swapping between the 2 versions of each chromosome that you already have before doing that to result in unique gametes. This is one of the many reasons that you can't have the same kid twice. Hopefully it helped, folks. We'll move on next time.